Today I presented of my laboratory study in my university. I want to talk with about study in my laboratory. Firstly, I said that about the plant tissue culture and produced in secondary metabolite by plant tissue culture. Plant tissue culture use of the product produced of the secondary metabolite in vitro condition. For that reason, we use of the explant of explant in part of the plants. We take off this explant and insert in the medium. This medium MS Murashing and Gamborg's B5 and neat medium. This medium use it in the plant tissue culture medium. We use of solid and liquid medium in plant tissue culture and produce it of the secondary metabolite. We use of the precursors and elicitor. In this medium, we increase on this secondary metabolite like of terpenoid, alkaloid, flavonoid, and other metabolites. After then we extract of this. Secondary metabolites in tissue medium. For that reason, we use of the organic solvent like of methanol, ethanol, and other solvent, ethyl acetate and hexane. Use of this solvent for extraction. We obtain of this. We obtain of the secondary metabolite. Also, we use of this clinical search, like of the antimicrobial and anti-cancer activities. We have got a lot of the many gram-negative and gram-positive bacteria use it in search. use it in search. This microorganism pathogen for human and other live things. We investigate of minimal inhibition concentration and disk diffusion method of the antimicrobial activities. We take often of this result and other clinical subclinical search. I am very sorry. Also, we search of the other clinical aim of the clinic anti-cancer activities. We have got a lot of the cancer cell line like of the CHCE, IT. IT 129 and other I am made it. I apologize all of participants for I have got one problem. I want that continue. I want that continue with research of the anti-cancer activity of this extract. We use of the anti-cancer activity VST1 method. This method is cell line incubated of the RPMI medium. We add of this medium fetal bovine serum, antibiotics, and RPMI cell medium. Also, we will add of the cancer cell line in well plate. One day in proliferate in this we line in the carbon carbon dioxide incubator. Overnight we use of the extract in the cancer cell line. After then we take off measurement of the ELISA reader. ELISA reader. We use of the 125 nanometer nanometer and measurement of the deed of the cell. We obtain of the anti-proliferative result, and we evaluate of the evaluate of the graph pet statistic program. Also, I want to talk about of my university laboratory. We have got a laboratory in my university. One laboratory plant tissue culture. We product of this laboratory plant micropropagation, callus propagation, suspension culture. Also, we increase of this method for secondary metabolite. We also Turkish have got a lot of endemic plants. This plant micropropagated of this laboratory every season, and every day we can produce this laboratory endemic plant or other plant. We obtain of this other plant, we search of the clinical research. I talk about of the scholarship about of my government. Firstly, researcher use of the Mevlana exchange program. This program supported by the Turkish government. Researcher and student use of this this scholarship program. Also, every time contact of my university international office. And other scholarship program, Erasmus Plus program. Turkey also supported of this program, but this program, especially European Union countries, supported by students and researchers for exchange. Also, we visit of the Poland in last year. And I stay in here. I stay in Poland. Your student, all of participant in the webinar use of this scholarship program. Also, my government supported of the other scholarship. Student follow this scholar program web page of the TÜBİTAK and High Council of Turkey High Council of Turkey web page. Thank you very much, all of the participants and listen to me. Also, I thank of my brother Dr. Akram for organizing this webinar.